Eukaryotic gene regulation is very complex, and it involves multiple regulatory elements that work together to control protein production. For example, enhancers, transcription factors, RNA splicing, and epigenetic control are all elements that contribute to eukaryotic gene regulation. Enhancers are a type of regulatory DNA sequence that are present in eukaryote cells, and the reason they are there is that eukaryote RNA polymerase cannot initiate transcription on its own. Enhancers are short DNA sequences that promote the process of transcription. Remember, transcription is the process by which DNA's instructions for producing proteins is copied by RNA polymerase to produce mRNA. Enhancers are binding sites for transcription factors, and transcription factors are proteins that bind to the promoter, such as the TATA box, and control transcription of the genes that they regulate. Sometimes the enhancer is a really long distance from the gene to be transcribed, so it has to be physically brought closer to that gene's promoter. Notice that in the diagram shown here, you see a DNA bending protein that is responsible for quite literally bending the DNA toward the promoter. This occurs so that the enhancer can make contact with the promoter. This then physically brings the transcription factors and other proteins responsible for initiating transcription closer together so that the enhancer can make contact with the promoter, so that the process of protein synthesis can begin. Transcription factors are proteins that bind to specific DNA sequences and other regulatory proteins. Some of them are activators that increase gene expression, and some are repressors that decrease gene expression. It is possible for two different genes to have the same promoter but have different enhancers and activators. This allows for differential gene expression, which can lead to the production of different phenotypes. The tumor suppressor gene p53 produces a protein that acts as a transcription factor. When tumor suppressor genes function normally, they are actively transcribed and produce proteins that regulate the cell cycle so that uncontrolled growth of cells does not occur. When the p53 gene is mutant, however, as it is in over 50% of all cancer types, the transcription factor produced by the expression of the gene can no longer properly regulate cell division. The role of p53 in cells is that it regulates the occurrence of apoptosis, or cell death. If this protein is mutant, apoptosis cannot occur normally, and cells can then grow and divide uncontrollably. This can lead to the development of cancer. Epigenetic control of gene expression also occurs in eukaryotes. The word epigenetic literally means to sit on top of genes. So what epigenetic control involves is factors external to the DNA that are controlling the expression of the genes in the DNA. This can happen when the histone proteins that eukaryote DNA winds around are either acetylated or methylated. The addition of either of these functional groups can result from diet, chemicals in the environment, environmental stressors, drugs, or even exposure to chemicals during development. The acetylation of histone proteins, meaning that an acetyl group gets added to the DNA, this loosens the DNA from the histone protein, making DNA more accessible for transcription by RNA polymerase. The addition of a methyl group or methylation of histone proteins tightens the DNA around the histone protein, making DNA less accessible by RNA polymerase for transcription. The result of epigenetic changes in DNA can vary from the development of cancerous cells to the development of autoimmune disorders such as lupus, diabetes, mental disorders, to name a few. Finally, alternative RNA splicing in eukaryote genomes is another way that gene expression is regulated. Remember that eukaryote DNA has introns and exons, and that the introns are non-coding sequences of DNA that are cut out, and that the exons are the express sequences of DNA that are spliced back together. Alternative splicing means that the exons are rearranged in multiple combinations to yield dozens of different possible proteins that can be produced. This mechanism makes it possible for a very few exons to encode for multiple proteins, so long as the exons are recombined in different ways. For example, the production of antibodies in the mammal immune system occurs as the result of alternative splicing of a handful of genes.
Gene regulation is tough, but you made it, so congratulations. Now let's talk about what you should have learned during this lesson. You should have learned that gene regulation is what makes it possible for cells to specialize in both structure and function. You also should have learned that gene regulation is what makes it possible for multicellularity and the complexity of organisms to exist. And finally, you should have learned that gene regulation is what makes it possible for the great diversity of life on our Earth to exist now. Now let's turn our attention to viruses, because they really turn the process of gene regulation on its head. Ready? Let's go!